Thank you very much, Ezer, for a very kind introduction of myself. As I said, that was my request to dedicate to this short talk about viruses and sedimentary systems, particularly focusing on carbonate settings to scientists and students in Turkey. Maybe I will do something, hide this panel. Okay, so as you can see in this first slide, Ezer already mentioned about some projects of mine which are focused on viruses and search for viruses in carbonate sedimentary systems. So I will show you maybe something which has been done in the last 2 years with my collaborators, and apart from that, I will show you some newest results published even I think last month or something like that. In the first slides, you can see two viruses typically found, two forms of viruses typically found in the natural environment, and one of them biologists call envelope virus. Enveloped virus is a virus which has a membrane which is pretty much a phospholipid bilayer. So it's a membrane which is built of phospholipids, a kind of fatty acids, and this membrane is attached to the virus itself from the host. In this case, envelope viruses are pretty much common in animals, but also eukaryotes. Apart from this membrane, which is around this, you see this having an interesting shape, fin, biologist called capsid, which simply contains genetic material. Okay, this genetic material can be either DNA or RNA, which is simply later transmitted, in fact, to the host cell, or simply a different host, which will be human, animal, or any living thing. More common in the natural systems is this form of a naked virus, which, as you can see, simply doesn't have this envelope, this membrane, and it simply contains a capsid, and I will tell you more about this capsid maybe later. If we have a fully developed, let's say, viron, I will introduce this term in the next slide. The capsid is connected with tail and tail fibers, and you will see what these two features are used for by a virus on the next slide. I don't know what's going on. Oh, here we go. Here we have an example of an infection process. As you know, viruses are not truly living organisms, and they, like bacteria or people, and they simply do not reproduce between themselves. They need a host cell to introduce their genetic material, DNA or RNA, through this tail, which simply pierces the outer membrane of the cell. Okay, so this is what this tail is needed for virus to infect our cells, for example, okay, or bacterial cells or any other cells. Virons are, in other words, these complete viruses which are living out of the cell, okay, outside the cell. They are moving freely in water and also in other sedimentary settings. Below, we have shown 3 microscopic images of viruses, or a virus. Some viruses are already inside the cell, or a virus which, as you can see, was caught in the process of infecting this bacterium. Okay, so here you can see this capsid, which has a specific shape. I will talk about it later, and a tail through which this genetic material is injected into the host cell. Okay, once this happens, the capsid floats off the bacterial cell and contributes to the total biomass. Soon after the infection gets started, genetic material simply mixes or takes lead over the genetic material of the host, and viruses start to replicate. As you can see, you have many of them here already in this cell. Eventually, there are too many, so that the cell bursts out and viruses are released and ready to infect again. These viruses, which infect a bacterium cell, are called bacteriophages. So the most common actually in the sedimentary environment. I will focus on them today. This is also here a very difficult maybe term to remember, but this process of which simply involves infection and replication and subsequent bursting out of viruses out of the bacterium cell is called cell lysis. Okay, next. And here we have a very nice helium-ion micrographic view of Escherichia coli. I will talk about this bacterium today also a lot. And as you can see, the bacteria are already infected by numerous viruses, all of them attached to the outer membrane of the bacterium cell through these, tail fibers. All of them actually have tails, you see, and they simply inject their genetic material into the host cell. Now, so we know generally about what viruses are and what to expect from viruses. As Ezra mentioned in the introduction, some viruses are good, some viruses are bad. We all know about it, but the viruses we work on are not that bad yet. Let's say we haven't found, Ebola virus or smallpox virus or something like that in our material. But the viruses we used are called, as I mentioned, bacteriophages. So is there a role for viruses in carbonate sedimentology? And here I will answer the question of one of professors who asked about whether viruses can live or survive in the snow or not. As you can see in this slide, viruses are very abundant. 10 billion per milliliter viruses are, and tens to hundreds more than bacteria. So obviously, this number gives us an idea that if there are so many viruses around, obviously they must do something. Okay, they also occur in sediments, especially in microbial mats, and I will show several pictures today showing viruses in microbial mats from Qatar and other places. Also, like bacteria, because they treat bacteria as their hosts, right? As I mentioned in the previous slide. So they live in all environments. It doesn't matter whether it's cold or hot. It doesn't matter whether it's snow, water, desert. Even they were suggested to be in the deep subsurface, perhaps even maybe on Mars, you know, who knows? Maybe some people say that this is very possible, that viruses can be found on Mars in the future. Of course, the limit for viruses to survive is temperature. It's temperature, whether it's, you know, cold or hot. Obviously, it's like with bacteria. It's a similar story. So in this case, they are contributing enormously to the biosphere as nutrient supply, food source, organic carbon recycling, and so on. And what I also wanted to mention, maybe some of you know it, but surprisingly, 8% of the human genome originated in viruses. So you can imagine viruses are very important for us, and they've been important for the Earth. In fact, we will see it also in one of the slides. So obviously, they must play a role. Viruses are usually tens to hundreds nanometers, so it's a very, very tiny object in diameter. The capsid, here you have it in a close-up view, the capsid is built of proteins. It can have different shapes, but the most commonly found shape in the natural environment is icosahedral. Icosahedral means 20 sides, 20 faces. It is also supposed that viruses can be negatively charged, just like bacteria, at neutral pH conditions due to so-called low value of isoelectric point, which is measured on viruses, and they can attract various cations. Here, of course, this includes calcium, magnesium, and others. Okay, so this is a good sign for us if we want to talk about potential mineralization of viruses and their role in the sedimentary systems. I don't know why it's stuck. And here is like a summary of environments where all viruses have been encountered on our Earth. And this is a study done 2 years ago already, where microbiologists studied around 200,000 various viral populations. You see their shapes here. Okay, some of them have tails, some are tailless. Okay, and they pretty much occur, as I already mentioned, in every climatic zone, on every latitude, and also at different depths. Particularly, there is some kind of variation of viruses in the polar regions. So, as you can see, we can potentially expect our viruses also in carbonate systems where the carbonates are being precipitated today. And the first approaches have been done on viruses and other minerals than calcium carbonate back in early 2000s. And here is one of such studies, experimental study, but also some data coming from the Rio Tinto copper mine in Spain, where we can see viruses, okay, with tails. Some of them have icosahedral shape, some are more oval, and as you can see, they are here attached to this iron precipitates, or some even are being permineralized. Okay, so that was the first suggestion that, or like for example here, look at this, that viruses can play some role in precipitation of various minerals. Another study comes from the siliceous settings, so where silica is precipitated, for example, in hot springs. Okay, also the study is experimental, And in natural system, I will show also some examples later. And here we have, again, this capsid, icosahedral in shape, and a tail, and here, very nicely permineralized viruses by silica. Look also at their scale. They are very, really tiny. The capsids themselves have less than 100 nanometers compared to bacteria, which are quite large. Okay. Now, so that my message for today is that there is a role, or we think there is a role, for viruses in providing nuclei for carbonate precipitation, like in the case of silica or iron, which I already mentioned. And they can be simply seeds or nucleation sites for such new minerals. The idea is not quite new. It was first suggested by Muriel Pacton in 2014, where he showed some permineralized viruses in microbial mats. Also, what well-known Bob Fogg called nano or dwarf bacteria in the 90s, we consider to them as most likely viruses. Okay, look at their shape, their size. We will also show some more similar pictures from microbial mats. This is a study from microbial mats in Brazil, two different independent studies, and here you have shown various viruses. Look also at their scale. Some of them are very nicely permineralized already. You see this darker colors. Some of them have different capsid shapes, and hence they were distinguished by names. You have them given in this slide here, which are actually the main families of viruses you can find in the natural systems. Some of them will have tails, some of them will not, and also, as I said, different shapes. Okay, someone wants to join our presentation. Okay, and now similar idea, but from the freshwater tufa, something what Ezer or people working in Palmucala, where travertines are being precipitated, would be interested in. This is our newest study currently in review in sedimentology. Here we have a bacterium, okay, which is surrounded by various viruses. Some have icosahedral shapes of capsids, some are ovoid. Look also at the scale of viruses, okay, around 200, maybe 300 nanometers, and look at the scale of this host. It's completely different, okay? They are ready, most likely, to infect the host cell. Again, our study from the Qatar microbial mats, it's a mesohaline system, so highly saline system, shows also a plethora of different viral-like particles, but also viruses themselves. And here I can show you, I'd like to show you two images. This is, both of them are transmission electron microscopic images, so we can give, so that we can give a very high magnification compared to traditional SEM approach. And here we have a bacterium which is surrounded by this darker EPS and most likely coccoid bacteria, and in the cell itself are tiny nanospheres of viruses, most likely we can tell waiting to burst out, so to lyse, so to kill, in other words, the bacterium and escape from the inside the cell. Next, we have a laboratory image of the same process, where a bacterium, have a look, it's filled up with numerous viruses, icosahedral in shape, compared with with this one. Here may be more spheroidal, which are also waiting to burst out. Okay, the process called lysis, just to remind all of you. And here is a very nice close-up picture of this helium ion microscopy, where, like in our Catarmat image, we have a very nicely recorded or caught virus in the action of infecting this bacterial cell. And I will show a very similar image later. Okay, here we go. So we have them here. This is a bacterial cell, okay? And most likely these nanospheric-like forms or icosahedral in shape. Look at the shapes. Most likely these are viruses which are attached to the outer membrane of this bacterium and ready to infect it. And here, look at the cluster of these nanospheres. Look at their also shapes. Okay, very likely to be viruses, and this will be very important for us when we will be talking about experimental approach. So bear in mind this image. We'll come back to it. Okay, and while talking about the experiments, which you know, some people in science completely forgot that something like an experiment might exist. We wanted to check the idea which was introduced by other scientists from different sedimentary systems, and simply we wanted to check whether calcium carbonate can also work with potential viruses or not. And we used what is commonly used, a Escherichia coli bacteria, which was infected by the so-called P1 phage, which is a bacterial virus, in this case, infecting Escherichia coli. So the size of E. coli is around 1 to 5 micrometers, whereas you see this broth, which was prepared by us, the broth of Bacteriophages, P1 phages, they are about 50 to 200 nanometers. So you can see how tiny they are. And here's a close-up view. This is a stem image where we can see this P1 bacteriophage build over icosahedral capsid and also a tail. We had a pure suspension, fat suspension, so we didn't have any bacterial cells. These bacterial pellets were removed. Also, no chemical impurities. As you know, Escherichia coli is a common bacterium which lives in stomachs, in our intestines, sewage, rivers, and lakes, and so on. Okay, and for our experiments, we used a solution, a carbonate solution. We simply added more and more sodium carbonate to the calcium chloride solution. Okay, and we did experiments with phages, and we also did, obviously, control experiments with no phages to compare whether there are any similarities between them or not. And here are the results of our experiments, which have been published finally, I don't remember, in November, I think, last year in GCA, where you can read more in detail about these experiments. And here we have carbonate particles with phages. So in other words, these are already permineralized viruses. Okay, we don't see viruses. We can only tell that they were probably viruses from their shapes. So do you remember the nanospheres I showed you 2 or 3 slides before? Okay, look at this material. Fantastic nanospheres of icosahedral or angular shapes. Some of them have dumbbells, very tiny particles, 40 to 60 nanometers. in diameter, and also, as I said, resembling permineralized capsids. Okay, what is also very important for us to mention, the bacteriophages we worked with and their precipitates have an ability to coalesce, okay, so to agglomerate. In other words, this is important for potential building of a wider area, which will be treated as a sheet for subsequent precipitation of a mineral, in this case calcium and carbon. These three images are SEM images. Below is a TEM image showing us 2 or 3 or 4 nearly attached, some of them already attached, some of them maybe will be attached later, permineralized icosahedral capsids of viruses. So as you can see, we have an evidence that likely viruses do influence carbonate precipitation. Moreover, in our control experiments, we only found a calcite. Okay, so control experiments, just to remind you, were without phages. Experiments with phages gave us 70% vaterite. Vaterite is a metastable polymorph of calcium carbonate, and 30% calcite. So we speculate, we hypothesize that vaterite, which lasted in our laboratory for the next 2 or 3 weeks, and it was still there, most likely is somehow stabilized. It's simply structure is stabilized by those phages. As this is something what needs to needs more work later. TEM images, just to show you the difference between control and experiments with phages. As you can see, we have some irregular forms of calcium carbonate and angular forms, icosahedral. Look again, it's fantastic. Some of them are already attached to each other. These are permineralized capsids of our bacteriophages. Okay. Now, something what I found out this month after we submitted this paper from the freshwater tufa environment. This is our experimental image showing attachment of these permineralized icosahedral viruses, and here we have a natural example, an equivalent of what was achieved experimentally. Look at this sausage-like structure, which has also icosahedral viruses already mineralized and attached to each other. Below you have a SAED, okay, a picture which analysis which gives us information that we have one mineral. In this case, this is a calcium carbonate. Okay, so perhaps our approach was not that bad at all. But coming back to the microbial mats, okay, we are talking about viruses which they are being mineralized and their influence on carbonate precipitation. The question is, could viruses be the seeds for crystal growth? Okay, we only now heard about possibility that viruses are permineralized, but what happens next? Okay, so look at these three images showing calcium carbonate crystals, okay, in Qatar microbial mats. In general, if you are familiar, obviously, with carbonate mineral precipitation, you know that we can have at least one type of precipitation, which is a so-called direct precipitation, which we call homogeneous or spontaneous nucleation of calcium carbonate or dolomite, like here, for example, in this image, which is not, let's say, only happens when the solution is supersaturated with calcium carbonate, okay? Whereas, so in this case, you need this supersaturated medium, okay, to precipitate calcium carbonate. Whereas the second way of calcium carbonate precipitation is through the heterogeneous nucleation, as we call it, okay? So in this case, which is much easier for a crystal to precipitate, is through a seed or a nucleation site or a nuclei simply, which we think are these tiny spots, okay, these tiny things, which are likely viruses because of their shape. So in this case, the seeds are provided by viral capsids, which this stage is biotic, okay? Whereas the precipitation of crystal itself happens in the abiotic stage, okay? So mineralized viruses are likely or give simply the nuclei for calcium carbonate precipitation, okay? It's a two-stage process. And recently, it has been suggested a similar approach, not experimental, but taken from the Hamlin pool stromatolites, very well known in the Shark Bay in Australia, where these authors suggested and proposed actually a model which presents or gives an idea about precipitation of this microbial mat, or which later becomes stromatolite or stromatolite itself, which is likely mediated by viruses. Simply, they are involved in the transition from a microbial mat to a stromatolite, okay? So why not? I think, confirmed it experimentally. But this is not everything we can learn about viruses. As you bear in mind, one of my first slides where I mentioned that 8% of the human genome is originates from viruses. Okay, obviously viruses may have existed back in the origins of Earth. Okay, so in other words, where first stromatolites or bacteria appeared, most likely viruses were already there. Okay, so we could say that the viruses were created in Charles Darwin's warm little pond, okay, and continued to evolve as other organisms do. Okay, either a bacteria or eukarya or archaea. We also have archaeal viruses. Okay, it doesn't matter. Viruses infect even fungi. Okay, so it's not a problem for them. They only need a host and nothing else. So we should find viruses in the fossil systems, and this is something what is very interesting, and I would like to focus on it right now. Okay. So let's come back to the pictures, experimental pictures, and also from microbial mats, showing these nanospheres of icosahedral or angular shapes, okay? And they are forming a sort of clusters, they are nicely aggregated. So to find fossil viruses in the fossil record, we suggest that the size of potential forms, viral forms, should have between 50 or 300 nanometers, or let's say, biologists say even 30 to 300 nanometers. They should be large numbers, similar in size, Also, you should look at their shape. Okay, nanospheres, icosahedral, particularly icosahedral, will be very easy to distinguish. But there is obviously a preservation issue. If there is a diagenesis, heavy diagenesis involved, that might be even impossible to find. But not always the situation is that bad for geologists, especially for young geologists who want to be on the top as soon as possible. Okay, so the context of finding potential viruses, of course, is with bacteria. If there are bacteria, there should be viruses as well, or if they, of course, are preserved, like, for example, stromatolites, like microbial mats or similar fossil systems. Mineralogy is a plethora of mineral forms, as I already showed you, but because we are focusing on carbonate systems, obviously this will be calcite, aragonite, or dolomite, but again, here we have some diagenetic processes involved, which simply destroy the original fabrics, and it will be difficult to recognize whether this tiny thing is really of viral origin. And what systems? Oh, here we go. Pamukkale, why not? Or an ancient equivalent of Pamukkale. So travertines, sinters, hot springs, and so on. Silica, phosphate, and other minerals. Here we have, for example, silicified viruses. Here we have an example from stromatolites. So viruses should be present throughout the geological record, and I will show you some examples. Maurice Taka already published very nice examples from the ancient systems, but I'd like to show you also some of them in my talk here. Here we have example from the Precambrian rocks, Paleoproterozoic. So it's, you see the age, it's quite old, almost 2 billion years old. And look at these tiny features. Okay, this is the scale. They are irregular. They are icosahedral. Many of these nanospheres. These are, could be silicified viruses. Okay, really tiny, 50-70 nanometers, and shape, as I already mentioned. The next example is from Paleoproterozoic phosphorite. Again, if we look at these tiny guys, okay, here. Again, nanospheres, irregular nanosphere. I would say, again, icosahedral capsids most likely. Are these phosphatized viruses? Okay, again, shape and similar size, say, okay, that could be, why not? The newly published paper from Libyan church, okay, of Messinian age. Again, authors suggested that these tiny nanosphere icosahedral particles are most likely mineralized viral aggregates. Here we go. Okay, so it's not that hard. People can still find them in the fossil record. And also, what about significance for viruses beyond microbial mats? That was the suggestion we made 2 or 3 years ago, and we wanted to obtain a sample of whiting. Whiting, as you know, is a periodic. It's a periodic, very massive production of calcium carbonate, particularly in the Bahamas, which is likely stimulated by dust, which is blown from the Sahara Desert over the Atlantic to the Bahamas, where simply these nutrients are used by bacteria, in this case cyanobacteria, which bloom very massively. And obviously, if they are cyanobacteria, they should be also viruses. So we wanted to check whether there is something related to viral precipitation, but we couldn't get this sample eventually because of the virus situation in the world. But it would be very interesting to check. Next is also pilloidal form or micropilloidal form. This example is from Neoproterozoic. We think that these microbial pilloids, which are composed of lime mud or micrite, you can call it as well, could be also viral pilloids. And again, if you look at these nanospheres, a cluster of nanospheres from microbial mats, here we go. Isn't it similar to what we have in the ancient system? Okay. So the pilloidal texture could be also the result of permineralization of viruses and bacteria. And finally, we reached the end, and I'd like to only summarize that viruses, as you imagine, were obviously neglected in herb sciences so far, but due to their huge numbers on Earth, obviously, we might expect that they could have played many roles, even back to the Precambrian itself, back to the origins of life. And obviously, viruses, I think I showed you some very good examples. We will continue this experimental work, but also collecting samples from natural carbonate systems. We think that they can be simply seeds for a carbonate precipitates. Okay, and here we have some examples of what I already focused in this short talk. Thank you very much for your attention. I think that will be all for now.